Boa tarde a todos, pessoal. Então, sejam bem-vindos à nossa última sessão de palestras, né, do módulo 4 da segunda SerCET 2021. Eu vou mudar agora para o inglês, tá, para poder apresentar os nossos palestrantes da Rice University dos Estados Unidos, tá certo? This afternoon at SerCET 2021, we're going to have Professor BETH BEASON-ABMAYR and Professor Scott Solomon, both from Rice University. Which title of the presentation will be Adapting Laboratory Courses to a Virtual Environment. So I will present Professor Beth's curriculum, which is, she is a teaching professor in the Department of Biosciences at Rice University in Houston, Texas, and a faculty fellow of the Rice Center for Teaching Excellence. She has developed multiple course-based undergraduate research experience and a student-centered integrative animal physiology course. Dr. Beason is a long-time judge for the International Genetically Engineering Machine, IGEM, competition and a member of the IGEM Executive Judging Committee. She is a past recipient of the George R. Brown Award for Superior Teaching and the Teaching Award for Excellence in Inquiry-Based Learning at Rice, and has published in Advances in Physiology Education and the Journal of Microbiology and Biology Education. A National Academies Education mentor in the life science, Dr. Beason is chair of the American Physiological Society's 2022 Institute of Teaching and Learning, and is an associate editor for Advances in Physiology Education. She earned a BS in microbiology from Auburn University and PhD in physiology and biophysics at the University of Alabama at Birmingham. So I would like to thank very much Professor Beth and Professor Scott for accepting our invitation to present here at SerCET, second SerCET 2021. So please, I will pass the word for Professor Beth. Thank you, Beth.
Thank you so much for that kind introduction, and I am very excited to be here today and share with you some of the things we did for our lab courses to adapt them to online learning. So I'm going to share my screen.
Just go to full screen. Yeah, it's okay.
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 So just to give you a little bit of background about Rice. So Rice University is an R1 institution, even though we're small, and we are literally smack dab across the street from the Texas Medical Center. So a lot of our students end up doing research in the Texas Medical Center. Most of our undergraduate majors participate in research at some point during their education. So to prepare them for independent research and also ensure that all of them have an enriching research experience while they're undergrads, we've transformed most of our undergraduate lab courses into research-based courses. So rather than just learning about science, our students actually learn to do science. And our undergraduate laboratory program in biosciences introduces, builds, and reinforces the research and communication skills our students need to become expert thinkers. All of our lab courses are team-based, and they emphasize the scientific process, not the content. So we're not so worried about them learning specific techniques. We want them to basically kind of understand how science works. And our lab courses also are unique standalone modules. So since they're not associated with a specific lecture course, like a lot of undergrad lab courses are, then we have a lot of freedom and flexibility with how we design those courses. When the pandemic hit in the middle of the spring 2020 semester, we were forced to shift to online instruction literally within like a week. And so at that time, we implemented emergency measures to ensure that our students were able to complete their work and also earn credit for those lab courses that they were taking that semester. This crisis prompted us to really reflect upon what we thought was important about teaching and try to come up with solutions that would promote student engagement and learning not just in a face-to-face environment, but in a virtual environment, which, as you are aware, has a lot of challenges, especially with a lab course. So today, each of us will share a story of how we adapted two very different types of lab courses to a remote online format. So the course I would like to share with you is I teach an advanced laboratory that's primarily for juniors and seniors in experimental synthetic biology. And the nice thing about this course is it gives a creative outlet for students to kind of explore what they're really interested in. And the course is kind of designed around three main themes where we spend a lot of time reading and presenting the literature, so the primary literature about synthetic biology. They also work as a team to design a circuit, and then they also gain experience with basically how you would put this circuit together. And for most of the students taking this course, they've just heard of synthetic biology, but they haven't ever really worked with it. So this is an exciting opportunity for them. So the course goals for this course, since it's a lab course, as you can imagine, a couple of the course goals, the ones that are highlighted in blue, are focused on lab skills. So actually, what kind of skills do you need to do to perform research? And then what do you do when you experience experiments that don't work out the way that you thought they would? The other goals associated with this course are more focused on scientific process and are not necessarily dependent on being in the lab itself. And incidentally, I actually use the same course learning goals for all of my lab courses for first-year students through upper-level senior students. So it's just the expectations for what they are going to achieve with each of those goals differs depending on which course they're in. For the fall 2021 semester, I decided to teach this lab course fully online. And I realized from the beginning there were going to be a couple of challenges, particularly would students still be able to achieve those course learning goals that were focused on lab work. So as I started redesigning the course, I started thinking about what were some ways that they still could acquire this even if they weren't physically in the lab. So I'm going to share with you kind of how the course was before COVID and then the changes I made after COVID when I adapted it to online. So before COVID, the class would choose one project that they would pursue throughout the semester. So each team would design a project, come up with an idea they wanted to work on, and then they basically would kind of pitch it to the rest of the class. And then the class would vote, and we would pick one project to move forward with. One of the primary reasons we had to do this is because of funds that it costs to support these projects. We just didn't have enough money in our budget to basically let every team pursue whatever they wanted to. So we had to kind of pick a project as a class for them to follow. After COVID, because now we no longer had constraints of money with supplies or for purchasing DNA, each team got to choose their own project. So they still shared it with the class, but instead of voting, the class gave them feedback to actually figure out how to make their project better. And what I learned from this experience is how important project ownership is for students to be engaged and interested in what they're doing in the lab. So when I asked them to kind of reflect upon their experiences in this virtual environment and what they felt like they learned, this was some of the feedback I got from the students. One student commented how difficult every part of the development of the project was, from basically coming up with the idea in the first place, designing an experiment, and then kind of figuring out what types of methods they would use if they were to pursue this project. And they've said that this is like the first time they've ever seen what happens on those initial stages. A lot of our students do research, but they basically join a lab that already has established projects going on, and they just kind of pick up where someone else has left off. So this gave them a chance to actually start at it from the front side. They liked being able to start really broad and pick their own topic and then narrow it down. And then one student commented how this was the first course that she had taken where she had the freedom to pick something based on her personal interests. So she was able, she's planning on going to medical school, and so she was able to basically tie in her interest in medicine into her project for this class. Before COVID, the students worked on design for the first 7 weeks, which is about the first half of our semester. And then in the second half of the semester, they went into the lab and they attempted to build their circuit. Mostly unsuccessfully, but they at least tried for 7 weeks to try to put it together. And one of the issues with this structure for the course is that the development of the project was really compressed in those first 3 weeks. So they hit the ground running and had to come up with an idea very quickly, share it with the rest of the class would vote, and then the class would design the DNA. So it was very, very heavy, skewed towards the very first part of the semester with coming up with the idea. And then at the end of the semester, they submitted an individual research progress report that described kind of what progress they'd made through the semester. After COVID, we spent the entire semester working on design. So the advantage of this was that students had multiple opportunities to reflect upon and refine their projects. So they didn't have to basically come up with the idea really fast, and then that's what they used for the whole semester. It actually just developed over the course of the semester, and basically we went through a iterative process where they would get feedback from me, they would refine their project, I'd give them more feedback, and we just and they got to share and present their work with their peers. And then at the end of the semester, instead of submitting a progress report, they submitted a team research proposal. So basically, everything they would need to do their project was submitted as in the form of a proposal. After COVID, it also gave us the opportunity to bring an expert in the field to give 6 guest lectures. So Dr. Carmela Haynes is a colleague of mine that I've known for many years through the IGEM competition. And she's at Emory University, which is in Atlanta, Georgia. Physically, she would not have been able to visit us for 6 times during the semester, but because we were meeting in Zoom, she actually would just join the class and basically share with them kind of from each stage of a project how you would actually develop this. So this was a very valuable addition to the class. Another thing we did after COVID is I had the students lead their own biotechnology review panel. So I had them watch a video from the National Science Foundation about how you review and score grants. And then the students built their own rubric based on intellectual merit and broader impact. And then they reviewed several projects that were just posted online on the IGEM website. And then we actually spent 3 days working on this panel where they would describe the idea and then strengths and weaknesses. And then they would actually assign it a priority score as if they were the funding agency. So this was a really valuable experience for them because it gave them a chance to critically evaluate the work of others. So if we go back to my original lab goals and my question, how could students still achieve their learning goals for lab work? When I was thinking about how could they still possess skills desired of an independent researcher, a couple of ways I thought they might meet this would be because they had to look at protocols, they had to think about methods, they had to think about how they would actually share that data. In their research proposal, they had to describe what experimental approaches they would take to build and test their circuit. You know, what are the DNA sequences? What would they measure as output? How would they measure it? What equipment and materials would they need? You know, how would they implement their system in the real world? So once they built this thing, how would they actually deploy it out in the real world? So all of this went into the research proposal. And I gave them a prompt on a reflection where I basically asked them, what is something specific that you did, observed, or learned that shows that you successfully accomplished one or more of the course learning goals? And one of the students said that he felt like he had achieved this learning goal about possessing laboratory skills because in order to put this proposal together and in order to design this project and plan everything that would happen, he had to really understand what methods they would need, what sort of data they would collect, how they would actually share that information. So it was much more than just, you know, saying we're going to do this. He had to really understand all the rationale. Similarly, for experience that not all scientific experiments worked as planned, I already mentioned how the students did the biotechnology panel, so they were able to actually look at the work of other students. But when I asked them this same question, almost half the class said that they felt like they had achieved this learning goal. And here's just a couple of examples. One student said this one was the most impactful, one of the most impactful learning goals for her, because their initial project idea changed over time as they experienced roadblocks. And so actually having to work through those roadblocks and think about how they would move forward really helped her achieve this learning goal. And then another student responded that when they wrote their paper for actually the design of their project, they realized that their initial project idea wasn't that good. So they basically scrapped their initial idea and started all over. So he felt like even though, even though he wasn't ever in the lab, that he still was able to experience this or achieve this learning goal. And at this time, I would like to turn the presentation over to my colleague, Scott Solomon, and he is going to talk about what he did with his courses. And then I'll join you back. I'll join back at the end to for some final reflections.
Oi, pessoal, vamos continuar essa apresentação. Now to continue this presentation, we're going to have Dr. Scott Solomon, also from the Rice University in Houston, United States. Dr. Scott Solomon is a biologist, professor, and science communicator. He teaches ecology, evolutionary biology, and scientific communication as an associate teaching professor at Rice University in Houston. He's also a member of the NSF, International Research. He was a fellow there at the NSF, at the Department of Entomology, at the Smithsonian Institution at Washington, D.C. He has been in Brazil as well, at the University Estadual Paulista, Unesp, in Rio Claro, not far from São Carlos, in fact. He has also worked at the University of Texas in Austin, where he got his bachelor degree, actually his bachelor in science in cell and structural biology. After that, he was at the Illinois at Urbana-Champaign. And he has also worked as, well, he is now in Houston, sorry. He teaches undergrad courses in introductory biology, laboratory courses in ecology and evolutionary biology, and a seminar course on public science communication. He has taught field biology courses in Texas, Belize, in Colorado, and has developed a complete digital lecture series on modern science and evolution with the great courses. Dr. Solomon's research examines the ecology and evolution of ants, and the coevolution between ants and microbes. He's a member of the American Association for Advancement of Science, the Society for the Study of Evolution, and he's also an associate editor for the journal Tropical Biology. So now I'll pass the word then to Dr. Scott. Please, you can assume from here.
Thank you so much for having me. And I'm going to see if I can get my slides to show here. I'm having some difficulties with getting my getting the connection here. So let me just try one more time if I can share. Yeah, I'm not able to share my slides. So Beth, I wonder if it's possible for you to, if you have my slides, if she can show those while I speak. I'm not sure if there's another good solution, but it's telling me that I don't have the ability to share my slides.
There's an application window there. You can actually share your screen from there.
Yeah, but I'm connected through my phone and through my computer.
Oh, I see.
My phone doesn't have the slides, so I have to
probably
share the
Beth
slides.
can actually show their slides.
Yeah, let me see. So Beth, do you still have a copy of my slides?
Yep. And then if you will just tell me when to advance.
Yeah,
because I'm gonna lose your video or your face when I share. So
I'll just let you know when I need to get the next slide. So while she's getting that pulled up, yeah. Oh, great. There we go. So so again, And can you hear me okay?
Yeah.
Okay, great. So thank you again for this opportunity. It's a treat for me to get to interact with you all because as you heard in my introduction, I've spent time in Brazil. I was in Rio Claro for about a year or so, and my wife is originally from Ribeirão Preto, and so we go back and visit family there as often as we can. And I've been to São Carlos many times. I have a good friend who's from São Carlos, and it's a lovely place, and it's a great honor to get to share some of my work with all of you. So as you heard from my colleague, Dr. Beason, we are teaching lab courses at Rice in a way that is a little bit non-traditional. And for the work that I do in ecology and evolutionary biology, one of the aspects that we really try to emphasize is getting students out of the classroom and out into natural ecosystems, where they're interacting with living organisms in their natural context. And as you can imagine, that poses a particular challenge for doing these lab courses in a virtual setting. So I want to talk a little bit about some of the ways that I've been adapting these courses to the virtual setting. So one of the classes that I teach is an introductory lab course in ecology and evolutionary biology. So this course is the course that students have to take before they move into the upper level, more advanced laboratory courses. And one thing that we always do in this lab is take students to a local natural area and have them measure the biological diversity of arthropods like insects and other little creatures that they can find living in the prairie and a forest ecosystem. And what they do is they take a collection of soil and leaves and bring it back to the lab, and they set those up in these contraptions here. This is called a Berlese funnel, and it's just a plastic soda bottle, so like a Coca-Cola bottle, that we've cut the top off of the lid and turned it upside down and put a light on top. And we put some alcohol, some ethanol in the bottom here. And the idea is that the insects will crawl down and become trapped and preserved in the alcohol. And then the students would normally go through those samples and use a microscope to try to identify what types of organisms they collected. And basically with that technique, they can then quantify the amount of biological diversity that they see in these different ecosystems. And so obviously we can't take students out into the field when we're doing this course in a virtual setting. So I had to really figure out, how do we achieve the most important learning objectives here while allowing students to do this type of activity wherever they happen to be. Some of our students are living on campus, but others are living at home in all different places all across the United States, and in fact in other countries as well, so around the world. So what, so Beth, you could go to the next slide now. So what I came up with is, rather than focusing on insects and other arthropods, we decided to have the students assess the biological diversity of the food that makes up our diets. So rather than sampling in a prairie or a forest, now the students are sampling wherever they have food stored. So this could be in their refrigerator or in a pantry, any place where they've got food. And we have them going through and doing a systematic survey where they will say, go to the second shelf and go to the third item from the left and select that item. And then they have to figure out, what are the ingredients within that food? So it's easiest if they can read a label. So if they have food where the ingredients are listed, they have to then look up, what is the organism that that ingredient came from? So with something like, rice, that's going to be easy. You just look up, what species is rice? But if you're working with something a bit more complex, like say bread, well, then you need to look up, okay, what's in the bread? Well, you've got the flour. What does that come from? It comes from wheat. So what species is wheat? What about the yeast? What species does that come from? And so they're figuring out what are the living things that make up our diets. And then they can apply the same methods for quantifying the diversity of those species as they would have done if they were looking at insects living in a prairie. So the question that we're asking here is a completely different question, but the tools and the approach that they're using is actually essentially the same in this example. And next slide. So I wonder if you can guess what was the most common species that they found in these surveys. This was sort of an interesting statement on what goes into our diets. Next, Beth. It turned out that the most common species that they found was sugarcane. In other words, sugar is the most common ingredient in the diets of our students. Probably not a huge surprise there. I think probably the students would have maybe guessed that in advance, but it was still interesting to see. So they found 427 different ingredients, 116 species total, and sugarcane is the most common of those. Next slide. Oops, you can go back one. Yep, thank you. Great. So that was an introductory lab course. I also teach an advanced laboratory course called biological diversity. And in this course, we kind of take that idea that you saw in the introductory class, and we spend an entire semester doing a deeper dive into the ways in which ecologists measure biological diversity in natural ecosystems. So normally in this course, what we would do is we would go out to a different natural area. This is a national preserve, the Big Thicket National Preserve in East Texas. And we spend the entire semester trying to quantify the biological diversity of different groups of organisms. And those include plants, fungi, reptiles, amphibians, birds, mammals, fish, all different organisms. And the students normally would come up with their own methodology. So they have to do some research to figure out what is the best way to survey fish, or what is the best way to survey and identify fungi or frogs. Whatever group that they are working with, they have to figure out how to do that type of survey. And then in the middle of the semester, we would normally go out and we would spend an entire weekend in this place and actually carry out the survey that the students have designed. And then what they would do normally is spend the rest of the semester analyzing the results of their survey and also sharing those results. You can go to the next slide, please. So one of the ways that we share the results of those surveys is through the website iNaturalist. So this is a website that anyone can access, and anyone can share observations of species that they've seen. So if you go on a hike and you see a particular type of bird, maybe you take a picture of the bird, maybe not, but if you are able to identify it, you can say, hey, I saw this species of bird at this place at this time. And then you can share that observation with the world through iNaturalist. So it's a really nice website because it allows people to share their observations, but they also can get feedback about whether those observations are in fact correct. You can go maybe to the next 2 or 3 slides. So you can see some pictures here. Next, where students have posted photos of different groups of organisms that they have seen in the surveys that they've done. And these are geotagged. So with their cameras, they're taking a picture. They can just use their cell phones for this as well. And it records the place and the time. And so what we get is this really nice database of different species that have been found in this particular region. And people will comment on whether they think the species identification is correct or if they want to propose a different species name. Generally, our students do pretty good at identifying these species correctly with some help from experts. We also normally share the results of these surveys with the National Park Service. So the organization that runs and maintains these parks is also interested in the results of the surveys that these students do. And I think this has been an important part of this class because the students know that they're not just turning in an assignment that their instructor, the professor, is going to read. They know that other people are actually going to be looking at the results of what they find. And I think that allows the students to really take ownership of the project, to take it seriously, and to see the value of the work that they're doing. Next slide. So clearly we couldn't do this this year in a virtual context. So what I did instead is I had the students go back and analyze the surveys that students have done in previous years. So they actually did a meta-analysis of the data collected by students over the past 7 years or so. And what we're able to do by doing that is actually look for trends in how the diversity of different groups of organisms has maybe changed over that time period. It also gives you a better sense of what is actually living out there by not just looking at a snapshot from one particular year, but over a number of years in sequence. So what you can see here is some examples of data from mammals. So you can see that the most common species of mammal is the deer, followed by a squirrel, and then another squirrel. And then there's a lot of rare species like our famous Texas armadillos. But when we looked at the diversity of these mammals and other species across years, in some cases, there were some interesting patterns. So if you look at the top right here, you can see how the diversity of mammals changed from year to year. And you can see that in 2017, there was a big drop in the diversity of mammals. And then it went up again the next time we surveyed mammals 2 years later in 2019. And in fact, what happened in 2017 is we had a big hurricane that struck the region, Hurricane Harvey, which led to some devastating flooding. It was a really, really bad storm. It caused flooding throughout Southeast Texas. A lot of people lost their homes, including myself. In fact, our house flooded and was destroyed. We had to rebuild the house. But for animals living out in the forest without any high place to go, this was also apparently a very devastating event. And we see that in the data because we see the decline in mammals in 2017. And when the students compared the diversity of mammals before Hurricane Harvey with the diversity after Hurricane Harvey, you can see in the bottom right graph, there was in fact a significant difference in the diversity of mammals, at least in the 1 year that we have data for after Harvey compared to all the years that we had before Harvey. So that was actually a really interesting thing that we couldn't get the normal way that we would do this class. We actually learned something by doing this meta-analysis that the students wouldn't have been able to learn by doing this the normal way. So there was actually kind of a neat advantage to doing it this way. Next slide. Another thing, I wanted to preserve and continue to have a way for the students to have their assignments for this class be used by people other than just their professor. So I wanted there to still be some product that the students produce that is useful to the broader community. And so what we did was I had the students develop some online resources. So they developed a website in which they shared information about some of the species that are found in the Big Thicket region. And so they developed this website. One of the things that they also did was to develop a field guide. So you can go to the next couple of slides here, Beth. So you've got some examples of, that's great. Examples here of, in addition to the website, they produced a PDF document that has information about a lot of the individual species that we know live in the Big Thicket, thanks to the surveys that the students have done. And the nice thing about this PDF is that we can print this out and make copies of it available at the park itself. And so one of the things that we're hoping to do is to be able to print enough copies so that this can be something that people can just pick up when they start going on a hike, and they can take it with them and use it as a field guide so that they can identify species that they see and learn a little bit about those species. So I think the students actually did a really nice job in developing these resources. And again, it's still something that they are contributing that isn't just a class assignment. It's something that lives on even once the class is done. Next slide. Another thing that we did in that class this year was to take advantage of the fact that we were meeting remotely to invite guest speakers to meet with the class and talk about different aspects of the work that they do that relates to biological diversity. So in much the same way that we are right now benefiting from being able to communicate across continents, and maybe we wouldn't have done this if we weren't all getting so used to having these virtual meetings. So in the same way, I invited a number of different guest speakers to come in and speak with the students about work that they do in quantifying biodiversity in different ecosystems around the world, work that they do related to the conservation of biological diversity in different places. And each of them works with a different group of organisms or a different part of the world, or has a focus more on different applications of what the students were learning about. So I think this was actually a really nice addition to the class, and one that I think we may try to preserve even once we go back to normal in-person classes again. Next slide. So to kind of wrap things up here, that concludes what I wanted to say about my specific classes. But what we thought we would do here at the end, and I'll invite Beth to join in as well with insights from her class, is to think about how each of our courses, the philosophies behind how we adapted each of our courses to a virtual environment, I think causes us to think about what is actually most important when we are designing classes in general. So we developed a series here of guiding principles that I think are useful in adapting these courses to a virtual environment. But I would argue that these are also useful just in designing courses for any context. So let me say a few things about kind of what we mean by each of these points here. And so the first thing is prioritizing supporting students. So one of the things that I've certainly come to appreciate in the last year is that we really need to make it clear to our students that we are here to help them and support them with their learning, yes, but also with what they're going through. And it's certainly been clear to me that the students appreciate this, and I think it really helps them to learn when they understand that they are being supported by their professors. So the ways that I've tried to do this include starting off the course by saying things like, hey, I understand this is a very, very challenging time, and I welcome your suggestions for what you think we can do to make this as valuable a learning experience as possible. So I try to make it clear that I'm doing my best, but I don't necessarily have all the answers, and I'm open to suggestions from them. I'm also really trying to go out of my way to meet with the students virtually, but one-on-one or in small groups, to try to get to know the students and to help them to recognize that that I'm really there to try to support them in whatever way I can. Beth, did you want to add anything about the prioritizing supporting students?
Yes. Can you hear me?
Yeah.
Okay. So to add to what Scott said, I learned the value of how important it was to really build a community. And this is much harder to do in a virtual environment than it is in person when we're meeting with them, face to face regularly. So trying to think of ways to, as Scott said, make sure that, we are aware of their needs and reaching out to them and finding ways to balance flexibility, expectations, and rigor. So not that we have to basically, change our learning goals so we don't, have the same level, but to basically be open to, thinking of different flexible ways we can be to help them achieve their goals. So varying the types of assignments like both Scott and I did, even dropping assignments that normally they would have done, and being flexible with things like due dates.
Yeah, that flexibility, I think, is a real key too, right? So, we've just had here in Texas, this last week has been a really extreme weather situation that you may have seen in the news. I understand it's been the international news what the Texas weather has been, because it's been so cold and we've lost power and water across much of the state. And so this has forced us to, once again, be very flexible in our due dates. We're extending the dates for many of our assignments, and I think it is very important when the students recognize that we're not just trying to force them to do work, that we really are understanding and want to allow them to do their best work. Oftentimes that means that it's going to be turned in much later than was originally planned. So the second point here, thinking broadly about learning goals. I think you saw a lot of examples of this in both Beth's presentation and in my own, where we really changed the nature of what the students are doing, but they still are able to get the most important learning goals out of these assignments. So for my, in my own case, right, switching from studying the diversity of arthropods in a prairie to the diversity of ingredients in our diet, it's a totally different question. But what is it that at the end of the day, I want my students to really get out of this lab? For me, it's much more important that they think about, how do we quantify diversity? How do we measure it? And how do we sample it in a way that actually is representative? It isn't biased in some way. And those same principles apply no matter whether it's food in our diets or whether it's arthropods in a prairie. So yes, they're missing out on some of the experiences and some of the techniques that they would learn in doing field work or in working with a microscope in my case. But I think they're much. I think it's much more important that they get those bigger picture goals of being able to quantify diversity. Beth, did you want to add something about learning goals for your courses?
Yeah, I think for my course, it was important for me to just recognize and accept that it wouldn't be possible for me to replicate online the exact same experience that they would have had in the lab. So it gave me an opportunity to focus on other learning goals and outcomes and try to think of creative ways that they could still achieve those. And I also tried to remember that this isn't the only lab course that they're going to take. They're going to have other opportunities to, as they progress throughout our program. And, as Scott said, it's not about the techniques. It's more about the way they think and how they work with things. And they really surprised me when they actually on their reflections picked two of the learning goals that I was thinking, how in the world are they ever going to achieve this when they're not in the lab? And they did. They were able to think about, like, oh, well, even though I didn't actually do this technique, I had to do a lot of research and understand how it would work and what I would need in order to do it. So that was very rewarding and reassuring to me that my students still felt like they were able to learn what we wanted them to learn.
So the third guiding principle is to give the students the resources that they need to succeed. And I think in some ways, this might be the most challenging aspect of switching to the remote format. At least it has been for me, because of course, normally we would give them resources in the classroom. They would come into the lab and we would be giving them pieces of equipment, or we would be giving them, tools that they would be using in order to complete an assignment. And of course, we can't do that in the remote format. So, I've had to be creative in my courses and trying to figure out ways that students can complete assignments on their own. One of the things that I've used quite a bit is websites and apps. So smartphone apps where students can download a free app that would allow them to do something like use their phone as a magnifying glass, or even like a small microscope. There's apps that allow students to measure things with their phones. So they might not have the tools that we would normally have in the lab, but they can use their smartphones to do many of the things that maybe not quite as well, but they can at least get the same general idea. Oh, we lost our slides.
You have something else in the slides?
Well, we just wanted to, I think, talk about these last points here. So points 4 and 5 here.
That's weird because it's still showing up. Let me stop and then try to reshare.
It was sharing, Beth.
Yeah, it was
okay.
Beth, it just reappeared and then disappeared just now. There we go. It's back.
Hi.
Perfect. Beth, did you want to say something about the resources for your classes?
Yes. So thinking about resources they might need other than, equipment or reagents for the lab, something as, making sure they know how to use our library to basically search for and access primary literature. So it's something that I just kind of assumed that they would be able to do that. But then I realized a lot of students didn't know that, they could log into our library and basically have access to all these things that are published. So making sure they know how to do that. Having them use Google Docs for collaborations and making sure they shared those with me so I could actually see as they're working on developing their project. And then we also took advantage for the virtual environment of using the website that's set up by the iGEM competition, because there are years of student projects with data, with slides, with information for them. And so basically using that as a resource for them to learn a lot about synthetic biology. And one of the things they really enjoyed is I had them pick their favorite iGEM project and share it with the class. And it was really funny because 2 students actually picked the exact same project. And but they gave a different way they presented it in a slightly different, emphasis and a different twist. So taking advantage, as Scott said, of things that are out there, either on the internet or on apps that they can use to actually still achieve those learning goals.
So the 4th guiding principle is to create learning experiences that motivate and inspire. And so, I mentioned in my advanced lab course that one of the things that we try to do is to create assignments that are not just for turning in and getting a grade, but they're creating products that are used by other people, by the National Park Service and by other researchers, potentially in the case of their iNaturalist observations. And I think that really does help to motivate the students to put a lot of effort into making these products as good as they can possibly be. And I think, the fact that they're doing research that is authentic. It is real research. We don't know what they're going to find when they go out and they do these surveys in the examples that I showed you. So I think that, and feedback from the students suggests that they really do get excited about doing this work when they know that it's not just an exercise that they're doing for a grade. They're actually doing this and they're solving a mystery, or they're collecting information, and they'll have answers that nobody else has. They get excited about that. And so trying to find ways in which we can preserve those aspects of these courses has been a really important guiding principle for me as I try to think about adapting these to online format. Beth?
Yeah, and for the lab course that I gave the example of, and also it applies to my other lab courses that are advanced as well as at the intro level, is giving students ownership of their learning is really crucial to basically having them be really interested and engaged in the project. And for the synthetic biology lab, I really did not give them I gave them feedback, but not until they had come up with the idea. And we started with, I had an assignment they worked on as a group where they basically just thought like, okay, if we could solve any problem in the world using synthetic biology, what would be local problems we wanted to solve? What would be national problems we wanted to solve? What would be global problems we wanted to solve? And that's where they started. And then they narrowed it down to an idea. And then they were like, okay, let's go see what's been done on this. And so basically giving them the chance to just, kind of go after what they're interested in and see like what it would take was extremely valuable. And, the advantage we had this semester with my doing it online is every group got to actually pursue what they were most interested in. Whereas before, when it had to be much more constrained because of resources, but money was not an option this time. And I basically told them they had an unlimited budget and, they needed to actually think about what it would cost, but basically, not to be worried about time or worried about money, to basically just think like, okay, what would it take for us to achieve this goal? And so I think that was extremely valuable for them to do that.
So the last of our guiding principles is to consider new opportunities. In other words, think about what it is that you may be able to do in the virtual format that you actually wouldn't be able to do using the traditional in-person format. And so as I mentioned in my classes, the main thing that I've been doing is to invite guest speakers and including guest speakers from all different parts of the world. And I really do think that has been a real advantage of the format. I think the students have gotten a lot out of those interactions in terms of learning and also in terms of networking, in terms of meeting people that they may develop some sort of a professional relationship with in the future. So this is something that I think, as each of us think about how we can take advantage of the online format, there may be things that you can do better online than what you'd be able to do in person. Beth?
Yeah, for similarly for me, the guest speaker was a extremely valuable part of this course. And even when we move back to in-person, I plan on inviting her to continue to interact with the class if she's interested in doing so. Another new opportunity was because I realized they weren't going to be able to get into the lab, was I took what would have been like a lab research project and turned it into a proposal. And many students said this was the first time they had ever been on that design end where they had to really think about what would go into actually achieving this. And so having to go through the whole process from what do we do research on, what's our question, to how would we actually make this in the lab, and what's the budget we would need in order to achieve this, was valuable for a lot of them. So some of them are going to grad school, many of them are interested in medical school, and so this was a different experience for them, being able to just think about that side. And I know when the semester started, some of the students were a little concerned because they thought it was kind of weird that they were, learning about synthetic biology without doing anything in the lab. And based on my experience with previous classes before this, they actually learned a lot more than when we did this before. I mean, just as an example, typically on their progress reports, I might get 3 to 5 references from the primary literature if I was lucky. For their research proposal, there were some teams that had 20 plus references all from the primary literature. So they actually really taught themselves, they learned a lot more about synthetic biology in this virtual environment. And that was something that I did not anticipate when we started, but I was really pleased with at the end.
Yeah, so that is basically, concludes the prepared remarks that we had. So we thought these guiding principles were things that both Beth and I found to be useful in thinking about adapting laboratory courses to a virtual environment, but also things that are generally helpful as guiding principles for developing any type of course. So we can go ahead and answer questions, I think. Do you want us to address some of the questions that are in the chat?
Yes. If you have finished, we would like to thank you very much for this very joyful presentation. And I think that the guidelines that you presented in the end are really, really important to guide us in the engineering and technological courses in order to try to do our lab classes. So it was really nice. So the idea now, if it's okay for you, we can start like a round table, a virtual round table, and Freddie and me, we are going to divide the questions for you. Okay? So first question: which kind of difficulties can the students have regarding online lab classes or courses?
So I think for me, what I've found is the biggest difficulty is it's much harder for me to see what the students are doing as they're doing it, and to try to intervene and make basically to identify what the problems are before they turn in their work, right? So normally we would be all together in the classroom or out in the field. They'd be doing something and I can go around and I can see what they're doing. I can ask them questions. They can ask me questions. And it's much more difficult for us to do that. So I'm seeing problems arise only after they've turned in some assignment. And it's harder to go back and figure out what went wrong early in the process. I think they also struggle from not being able to work together as easily, and it's the same kind of an issue there.
Yeah, my students had similar difficulties. What helped is because we were using Zoom for our classes, then I was able to put them in breakout rooms so they were able to interact with their small team. And then what a lot of my students did is they just kind of set up their own virtual meetings outside of scheduled class time. And so that way they were still able to interact. But I wasn't able to, but the breakout rooms in Zoom are really nice and I could still go visit with each team. But what was lost was kind of, you know, visiting with everyone all at once in the big space where then I'd hear like, oh, they have a great idea. Let me share this with the class, right? So it was much harder to do virtually, because then when we were meeting as whole class, we got a little bit of that back, but it wasn't quite as natural. It was more like, you know, they almost had to really think about what they were going to say in front of the class. So it was a little bit awkward. Not there was no the spontaneity was lost. So kind of the excitement when they're just like the light bulb goes on in their head and they're like, oh, I have a great idea, I want to tell the whole class. That was kind of lost in the virtual environment. And then another challenge, I think, is basically the student's mindset of basically seeing that just because the class is online doesn't mean it's not a lab, and it doesn't mean that just because they're not going to be, you know, picking up a pipetter and, you know, or growing a cell or going out in the field and collecting a sample, it doesn't mean they're not still achieving important learning goals. And so I think that's getting that past that kind of like mental block in the students took us probably a couple of weeks. So then once they realized like, oh, this is a lab and I am learning, then it got a lot easier. So.
Okay. Fred?
Well, it's very interesting. Very interesting to hear from you that the students are actually learning more than in the present laboratory. That's quite a surprise to us, and would like you to comment on that. But before you comment on that, we have another question here concerning the material you have used in this experiment, remote experiment in your teaching laboratory. Did you use some sort of, you know, videos from previous experiments from laboratories from your university or other universities for the students?
So I did not in my course because I shifted the focus to be more kind of the design and development. I do have a colleague at Rice who is teaching a microbiology lab, and he's also teaching an electrophysiology lab. And he makes recordings of him actually doing the experiments and shares those with his students. So they don't get the hands-on experience, but they are able to see him do that. And I know there are a lot of universities that are recording things. And you know, and my students, my TAs actually, you know, offered to make some videos if we needed them. So we do have the cameras and stuff to do that. I just haven't done it for my classes, but my colleague has. Scott.
Yeah, I think what we've had to figure out is how important are the techniques that students would learn in each of these classes. And in some cases, like in the colleagues that Beth was mentioning, the techniques really are an important part of the class. That's one of the important learning objectives, is to know how to do these laboratory techniques. And then you have to figure out how can the students learn those techniques when they're not doing them themselves. In my classes, I decided that the techniques were not what was so important. And that, you know, if they go on and take other courses in the future, maybe they can learn some of those techniques. But I felt it was much more important to focus on the thinking like a scientist, right? Thinking about how to design an experiment, analyze data, interpret data, communicate results. Those aspects for my classes were more important than the techniques themselves. So I just decided I'm not going to worry about trying to teach these students the techniques that they would be learning. We'll allow them to do that in the future in some other classes that hopefully they can take in person.
Yeah, that was similarly for my class. We actually focused on, I think throughout the course of the semester, they gave maybe at least four presentations. Some of them were really short. A couple of them were team presentations. They had to do an individual presentation. So they still were talking about science data and sharing it and communicating it. But that, I really shifted the focus to be more design and then how you would communicate that, as opposed to how you would actually do it.
Do I do this one, Fred?
Yeah. I'll do the next one.
Do you think it's worth it to use work groups in online lab classes, or just individually it's possible? What do you think about it?
So most of our lab courses at Rice are all team-based, and I kept the team-based aspect, and that was essential. So when I actually asked them to give me feedback when they were preparing their proposal, and also when they were preparing presentations, and kind of tell me like who contributed what. And there were some people that were really felt really comfortable looking up a method they'd never done and basically digging around and see like, oh, how would we actually do this, right? There were other people that actually looked up the background information. And so keeping that team focus, and these are small teams. My teams are like 3 to 4 students. So keeping that small group together made for a more enriching experience for all of them. They give joint presentations. So they're, it's the individual part, I think, would have been a lot more stressful. In a normal lab environment, the individual part would be stressful, and especially in this virtual environment, I think it would have been even more, because then they would have been doing all this work just themselves. But by having a team-based approach, they were able to kind of share the workload. So I think that was crucial for at least for my classes.
And I've done it both ways, actually. So in my introductory lab course, the students would normally work in groups on most of their assignments, but I decided that it was more important for me to give them flexibility right now, to allow them to complete the assignments whenever they want and wherever they want. And I felt that if I was going to have them work in groups, it would remove some of that flexibility. It would make it harder for them to really take advantage of the extended time period I'm giving them, and the flexibility to do the work where they want as well. So in that introductory course, I'm having the students just work individually on all of the assignments. In my advanced lab course, the biodiversity lab, we are continuing to do group work. And I think that for the most part, that has been a good thing. I will note, though, that in at least one case, I'm aware that there was a group that had some real problems. And that happens, right? Anybody that's taught classes with group work knows that often there can be some conflicts between individual group members, even just under normal circumstances. But I think that it's exaggerated now. And without going into too many details, I think what happened in this case was one of the students in one of the groups had some kind of emergencies or crises that were happening with him or his family, and he wasn't able to participate the way that he normally would. And he communicated that to me, but he didn't communicate it very well to his group members. And so they got frustrated that he wasn't doing what he would normally be doing. He felt bad, and it didn't, you know, really get to have the same experience. I was trying to respect the student's privacy, support him, but also help everybody to complete the assignment. And so I think it added a lot of stress in the case of that one group that I don't know in hindsight if I think it would have been better to not do the group assignment or maybe to intervene a little bit more. But I just point out that I think all of the challenges of group work still exist in a virtual format, but they may be exaggerated because not just because of the group, not just because of the remote format, but because so many people are just going through a lot of stress and have other things going up, and they're not always comfortable sharing what about the details of all of those things.
Yeah, very interesting. Well, the next question is you might have already comments on the answer for the next question. Which is about assessment. How you are actually doing the assessments for individual students? I believe that they are working remotely among themselves as well when I'm talking about the groups. And how, and for complementing, how can you say, or how did you measure the evolution, individual evolution, and why can you say, why are you saying that, you know, they're learning more than in the present, in the presential, you know, mode?
So for my class, yes, I imagine most of the groups were working remotely. Some of them were on campus, so they might have met up, but it was probably easier for them to meet remotely. As far as individual progress, a lot of this course was team-based products, so team-based assessment, but a huge component of all of my courses are individual reflections, where I basically want them to basically think about what they've learned, how they're learning, you know, what are they struggling with, and to give me feedback about that. And so I have multiple individual those types of assessments throughout the semester. And so I'm able to see from their feedback, you know, kind of how they're making progress and what their level of understanding is. And additionally, even with the team assignments, as I said, they had to basically tell me like who worked on what part. And so then I would be able to see if they were struggling with a certain, you know, aspect of the project. And then when they were giving presentations in class, those were, you know, I saw the slides they produced. I saw, you know, were they able to answer questions from me and the classmates? And so the assessment actually for the online for my course was not that much different than what it would be in person. I was able to shift a lot of this stuff online. And then to answer the question about, you know, how did I think they learned more? It was because the level of detail and depth and explanation they gave, the design of their circuits, like now compared to what it was just last spring, was like night and day. I mean, they were able to answer questions that they'd never even thought about before. You know, even to the extent of, oh, if we wanted to actually, you know, do this in the real world, we would have to be concerned that not everyone could afford this. And so we would have to think about, you know, what would be a cost-effective way to do it. We can't just release this out into the environment. So here's our design. These are not engineering students. These are bioscience students. And they in their proposals, they were showing me designs of how they would actually have a self-contained system that would actually be put into the environment to actually deliver their product. So they were thinking in ways that they haven't thought before. So it was actually going way beyond just, here's what I want to build, now let me work on it. So the products that they, you know, throughout the semester, the products that they shared actually helped show me that. And then the other thing I added, which we had not done before, was the they had to evaluate the work of others that was in this field. And so they had to really think about what makes this a good project. You know, is it scientifically sound and valid? If I was giving you money, would I support this? And okay, yeah, this is good science, but what's the impact on society and kind of the world as a whole? So they had to actually think beyond the bench. And so I think they got a much more in-depth, broader perspective by working on it this way than they had in the past.
Mm-hmm.
Yeah, for me, you know, I'm not sure I really have a way of comparing the outcomes of the virtual labs versus the in-person labs in any quantifiable way, but you know, I do survey the students every semester and ask them for their feedback, in addition to the normal surveys that the university asked them to complete. And the students were actually surprisingly positive in their feedback about how they felt the classes went. And I don't know quite what to make of that. I think maybe in some ways it's just their expectations were very low. I don't think they expected these classes to be very good at all. And so the fact that they, you know, weren't terrible, I guess, means that the students were happy. But in terms of how I evaluate individual students in a group context, you know, I always ask the students to evaluate their own, to do a self-reflection where they evaluate their own contributions, and I ask them to evaluate the contributions of their team members, and then I compare those. And so I normally do that even when we're working in person, and we did the same thing here. And what I find is usually they match up pretty well, right? So usually the students, if there's somebody who maybe isn't really pulling their own weight, they usually admit to that. And then their teammates are usually tend to be kind of nice to them and don't rate the person who wasn't pulling their weight too low, but it shows up as lower than what it would otherwise be. And if there's ever any question about whether something went wrong or was there a big fight that happened and people are being unfair, then I can go in and I can start actually talking to the students and finding out what went wrong. But usually that's not necessary.
And something that I was just going to add to comment on Scott's talking about they're evaluating each other. So something that I have my students do in all of my lab courses, and we were doing this pre-COVID, and so it actually, you know, is as applicable regardless of whether it's virtual or face-to-face, is at the very beginning of the semester, after they know, like I assign the team so they know which team they're on. I have each team create their own group contract or team agreement where basically they lay out how they're going to work as a group. So how are they going to meet? What do they, you know, how are they going to work on assignments that are team-based assignments? How do they prepare for lab? What happens if there's a conflict? How do they resolve it? You know, when do they come to me? Or, you know, and so each group does their own. And then I basically, throughout the semester, remind them, all right, you know, go back and look at your contract, and or is each group member kind of holding up to that contract? And so since I started having them do that, then the group work has actually really improved, because now they all have to sit there and really think about, you know, what does it mean to be, you know, a contributing member of the group and to contribute equally. You know, it kind of holds them accountable. Because they basically have laid out for themselves, this is how we're going to work together as a group. And there's still bumps and there's still hiccups along the way, but usually they are able to resolve those issues themselves, and only rarely does it escalate to where they have to come to me and say, we need help because we have someone that is not contributing. And so I think this was even more valuable in the online environment. I mean, they even put into their contracts, like, you know, what happens if we lose internet? You know, we will make sure we have multiple ways to reach out to each other. And so I think it actually was very valuable for them to think about, you know, in this new learning environment, how they were actually going to work together. So
okay, I think with this we are ending our session, our lunch session here in São Carlos, here in Brazil. Because we have something like 10 or 15 minutes to start the afternoon session, okay? That we're going to do a live problem learn class, okay? So we're going to do like a Big Brother live class. So we have 10 or 15 minutes. And I would like to thank you, both of you, very much to accept our invitation in the name of the organizers. And we had to struggle a little bit with this change from last Thursday to Monday, but thankfully we could do this presentation of you, which was very, very rich for us. I wrote down a lot of ideas for our lab classes, and I hope that we can use your ideas here. Okay, if Fred wants to do some final comments, and I would like to just to finish, I would like to have some final words of Scott in Portuguese, if possible. Okay, Scott. So Fred, and then Scott, please.
Well, I'd like to thank you both as well. It was really interesting for me to learn your experiences, and I'm sure our audience will use most of the ideas, particularly the guiding principles for remote labs. This was quite a very nice tip, I would say. I believe that you have discussed that together and you agreed on that, and we're going to do the same over here. And yeah, I'll pass it on to you, and thanks again.
Ok, muito obrigado para todos. Foi prazer e boa sorte. Muito obrigado.
Okay, so bye. See you.
Ciao.
Thank you so much.
Thank you.
Bye.
Augusto, posso dar um último recado? Para a gente avisar o pessoal que vai assistir as palestras que nós vamos encerrar essa sessão. Então, quem quiser assistir a sessão a partir das 14 horas, tem que entrar no site na sessão sexta-feira à tarde. Quer dizer, vai entrar em outra sala para poder assistir a sessão da oficina. Eu acho que era isso, né, Fred?
Isso.
Tá legal. Segunda à tarde, né? Sessão de segunda à tarde. Obrigado. Obrigado, Augusto.